Hello guys, my name is Dr. Varun and you are watching Chlorophyll classes only on YouTube, where I will try to make your concepts of biology super easy. Well, we are preparing for NEET 2026, but it doesn't mean that the students who are gonna give their paper in 2027 should not refer to this page or this channel. They can. Okay, meanwhile today we're going to be studying about the 12th class syllabus, which could be coming as an important topic in the boards also, as well as for your NEET also. So let's just dive into it. Now what it is our today's topic. Today we're going to be studying about, like, if you know that what we were covering about till now, we have covered about that non-allelic inheritance or effects or expressions. Basically the genes which are not like for example genes, one gene has alleles but other genes are also interfering and working with it, right? This thing we were studying in the last. This is what little bit more to that also only. Okay, so means these are more than one genes when are working. Now inheritance on this basis could be of two types till now what I have discussed. Earlier when we were studying about the Mendel genetics, it is basically about what? It is about that how one gene is affecting the character. So means there is monogenic inheritance. So one thing which we will be studying today is what? Monogenic inheritance. So what is the monogenic inheritance? What we have studied in the Mendel. Okay, so if I have to write it down, what I will say that one gene is responsible for one character only. Like for example, there was a gene for what? Height, stem height, or seed color, seed shape, right? So this is monogenic inheritance. Whereas another one is what? Polygenic inheritance. Polygenic means, poly word itself means what? Many genic gene. Okay, many genes inheritance and how it is affecting one character. Okay, so now you will be like, sir, how come one character is being taken care by more than one gene? It happens in case of humans, one character that we're going to be studying about it. Okay, so polygenic inheritance, which is talking about that more than one gene is responsible for the expression of one character. This is super important for your boards. Okay, and super important, of course, its difference and understanding the concepts and statement for your NEET examinations. Okay, so now you have to remember that poly means multiple genes are working. Multiple genes means for one character more thing is required, means quantity. Okay, so here what happened, as the quantity increasing, the expression also intensified. Okay, so means what? That if, for example, if something is doing particular thing, for example, if height, let's take an example, it's not there but we are taking example. So if a person is 5 feet height, okay, so what happened after certain, if I add one extra gene, it will be 5.2, one more extra 5.4. so what happened? Quantity increases, expression of the character also increased. Okay, expression doesn't changed. In the last video about the inheritance, we were saying what? Expressions are changing up. Here the expression is quantifying. Okay, so means here can we say that polygenic inheritance is what? Quantitative inheritance. Okay, so it is what? A quantitative inheritance. So if a question came that give an example of quantitative inheritance, polygenic inheritance examples. Okay, remember it. So it is what? Quantitative inheritance. But what happened over here in monogenic inheritance? It's about either recessive or the dominant. Even if single T comes or double T comes, expression remains what? Tall. Please pay attention. So monogenic inheritance, one character, even if two, it comes in homozygous condition or heterozygous condition, the expression remains same. Okay, so this is what it is, quality. Single also, double also, quality remains same. It won't change the expression or intensify the expression. So it is what monogenic inheritance is, qualitative inheritance. Very important for you, beta. Just remember this thing, qualitative inheritance. Okay, let's just dive into polygenic inheritance because that's in our main topic for today. So polygenic inheritance. So some characters are influenced by more than one genes and they intensified its effects. Okay, as the more dominant gene is coming up or the allele is coming up. So this is what polygenic inheritance. So this is how we're going to be right. Some characters are controlled by more than one gene, and addition to each will do what? Intensify the expression. And addition to every dominant allele will intensify the expression of the character. Example which you have to remember is the skin color. So you know that there are so many shades of skin color. There might be white people, there might be black people, there might be intermediate brown people, there might be dark brown, light fair. These colors of shades are there. How they are coming up? They are coming up because the skin color is taken care by 3 different genes. So as the quantity increases, their expression also becomes darker. Let's just try to understand with the examples because that is how we learn better. Okay, so in the polygenic inheritance, for example, skin color. The first example we are taking about skin color. Later we will take one more example in the plants also. So that if it comes that given example with respect to plants in board exam and with respect to animals, you have option. Okay, so this question can come to you. Okay, so now skin color in human is controlled by 3 different genes. That is, let's take it A, B and C. Okay, now we are doing what? We are doing a cross between homozygous dominant and homozygous recessive form of it. So recessive means small, dominant means all plus. Okay, so a cross between homozygous dominant, that is capital A, capital B and capital C, with a homozygous recessive parent, that is all are small, will have following types of progeny. Let's just make this cross. This cross is also what? A trihybrid cross, which we have not studied before, but here we are doing it. Of course, if you go to any coaching institute, they will teach you after the dihybrid cross. But I don't want to waste your time over there, so we are just directly jumping up over here. Of course, for tips and tricks, you can go to either the shorts topic of it, or you can go directly to what? Towards the ender topics when we will be studying about the tips and tricks more. Okay. So now, so let's make it. So one parent is what? Dominant. see, I'm writing it like this so that I can distinguish. Okay, and the recessive parent is like this. So now how they look like? Now the capitals one, the dominant parent, will have a dark black color, and in terms of science we use the word as negro. Okay, so this parent would be negro. Of course the negro word is racist and it is banned to use, but it is given in the books. Okay, darker color. Okay, and the smaller ones are what? They are albino. We use the word albino, means color is fairly white, no shades are there. Okay, currently my body has certain shade. Okay, your body has certain shades. Let's just hype into it. Let's just see. So if they will make gametes, what are the types of gametes they will make? So these are the parents, and the gametes would be as follow. So their gametes all would be the capital ones. So A, B and C. Here small ones, these would be the gametes. Now what will happen? These gametes will, well, there will be a cross. So there will be fertilization, and the zygote that will form will have what? It will have capital A, capital B, small v, capital C, small c. Now this child that will form, it is intermediate, means it will be dark white mix, that is fairly in between shade of it. Okay, so the shade would be what? Let it intermediate. Now the problem comes. What is the problem? That, sir, where is the shade? Okay, but a shade will come. How they will come? Because of this intermediate. So what happened? Think about that. A black guy from a black skinned guy married a fairly white skinned girl. Okay, and then they had a baby. The baby will of intermediate shades. Okay, and that intermediate shade is what? In between black and white, that is the brown shade we say it. Okay, so this is intermediate shade. Now when this intermediate same color individual for like gametes form, then what happened? Then F2 generation will form. That is only what we're gonna be seeing, and see that how the different shades can come up now. Okay, so now what happened? Now we will be doing what? Selfing of it. Selfing. Now here, if you have been with me since the beginning, you know that what all gametes it will make and how many types of gametes it will make. So tell me that how many types of gametes it will make. First question. Pause it and then write it in the comment section, or maybe just write it at your place and just see that whether it's correct or not. According to me, the formula for the number of gamete is 2 raised to power n. Okay, that is n is what? The heterozygous condition. Here there are what? 3 genes are in the heterozygous conditions. Okay, so 2 raised to power 3, that is 2 into 2 into 2, that is 8. 8 different types of gametes will form. If you don't know what are the types of it, you will use Forklin method to do it. Okay, so with the Forklin method, what would be the gametes? I'm going to be writing up. So I am making the Punnett square directly to it, and 8 gametes of female and the males over here. Okay, so let's just dive into it. This is F2 generation. So 8 gametes means 8 row and 8 column we have to make. Same here. Let's see the gametes by a Falkland method. Are you able to make it? If you are able to make it, let's just see that whether they are the correct ones or not. Okay, so the first gamete, that would be male and female gametes. Okay, the first one, A, B, C. Then comes A, B, capital C. Okay, let's just make it. I also make it over here. So A, so A, B, capital, A, B, small c. Okay, so I will write it as such. Then A, small b, capital C, A, small, small. Then small a, capital B, capital C, A, B and C. Then small, small, capital, small, small, small. Okay, so same gametes would be of the other side. So this is the other side. Now let's just write it about them. So I am writing it with the yellow color. So quickly we will see it. So capital, all capitals are there. Okay, then we have capital, capital and small. Capital. So it will take a little bit of time. So let's just pause it and I completely fill it up. Okay. So here are our whole completely filled video box Punnett square would be. Now what you have to see in it. Okay, so you have, it's ideal if you do it yourself. Okay, so I'm just giving you the hint and then I will give you the final answer. Let's just see that what is happening, that over here you have to find that how many capitals are there. Don't see that A, B, C. It should be either the capitals or the small alphabets. So the ones which are like, you have to calculate that there are 5 capital, 5, so 5 dominant, 4 dominant, 1 dominant, 0 dominant, like this way. Like for example, the first one is what? If there are 6 capitals present. So 6 capital is present only in this case. Can you see that? So means 6 dominant characters are there. So I would be writing it on a next page. That is what? 6 dominant alleles present. And if you will see that total how many present? Only 1. Okay, so that would be what? 1, and the color of 6 dominant would be what? Negro. Let's just go back. Now we comes to the point that there should be 5 dominant alleles, means 5 capitals present. If we have to find the 5 capitals, we will find that if you see that 1, 2, like over here, 1, 2, 3, 4, 5. So this is what? This is a dominant one, 5 dominant one. Okay, same goes here, 5 dominant one. Okay, can we find more than this? Let's just see over here somewhere if we can see. Look over here, this only 1 is small, so this is also. Okay, let's see more if we can find it out. This is there, 4. Okay, this. Okay, 4. Anything else would be there? Let's just find out quickly. This is there. Okay, anything more than this? I think one more capital C is capital B is there, capital A is there, one more C would be there. This, only this. So how many were there? 1, 2, like 1, 2, 3, 4, 5, 6. So 6 are what? Which has only 5 dominant. So next come what? Are 5 dominant alleles, because maximum can be 6 only. All of them are capital, right? So 5 dominant alleles. Same, if you have to find that they were what? 6 would be there. Then 4 dominant alleles. Then comes 3 dominant alleles. 2 dominant alleles, 1 dominant allele, rest all would be smaller, okay, and 0 dominant allele. So these all options could be there. 0 dominant allele would be what? Your same single one, okay. So that is what the albino or the white. So this is one, that is your albino shade or white, which will only be one if you will find out in the text. Same, let's see that how many of them they are there. If you are able to do it yourself, very good. Let's just try to see that. If you have not, stop this video, make it yourself, find it yourself, and then come back to this answer. Let's just see that the dominant alleles, with the 4 dominant alleles, it will be R15, this will be R20, this will be 15, this will be 6. So if you see that once you row one range, the other range would be like this. This is the magic of maths which is happening over here, I will say that. Now 3 dominant alleles, one will look like what? Intermediate, if you remember. Now what will happen? This one, from going to here, the shade will become darker. So here it will be darker shade. And from here to here would be what? Lighter shade. So it is just like what? Fair and Lovely ad. That Fair and Lovely, use it for this much of a time, the shade becomes darker or lighter. All is bullshit. Okay, let's just come back that it basically feature that is resembling to this. Okay, so what will happen then? As we are going towards intermediate to negro, the colors would be what? Fairly black, it will be dark, so it will be what? Fairly white, it would be light, and this. So this is how it will look like. Now here the most important thing is that now in paper we can even make the graph of it. So if you have studied in 10th class stats, in the stats you used to make the cumulative graph. Okay, so the cumulative graph can be asked to you. Before going to it, can we write this? This is what? Phenotypic expression. Phenotypic expression. So what would be the phenotypic ratio over here? Phenotypic ratio, it will be what? 1:6:15, ratio 20, ratio 15, ratio 6, ratio 1. So this also you have to remember. Plus we were talking about the cumulative graph expression. Cumulative graph expression of this. How it will look like? It will look something like this. So there is what? X-axis, sorry, y-axis and the x-axis. Okay. So here it is what? Here you are telling the number of phenotype. Number of phenotype. Here you are showing what? Shade. Okay. So starting with the first one, that is what? Your negro one. Okay. This one. So it is what? 1. So let's say that this 1 is represented by this box. Okay, so this is 1. Then comes 6. So it will be represented by this, let's say this 6. Then it comes what? 15. So this is 15. Then 20 would be what? Further higher. Okay, so this is what? 20. Then again comes what? 15. So 15. Then again comes what? 6. So like this. 6, and the last one is our negro, that is 1. So this is what? A graphical representation of it. Further more variety of it we used like this way. So if this graph comes like this, is what? Showing you what? How to expression. This expression is what? A polygenic inheritance expression graph. Okay, I hope you will remember it. This can be asked to you. Let's just say that I told you what? It has one more example which links to what? It links to the plant. That is what? Kernel color in the wheat. Second example, that is kernel color of wheat. Actually they are brown in color, but we don't say it brown, we call it red. Okay, so kernel color in wheat. So they also show the same expression. Okay, so kernel color is expressed by what? It's a polygenic inheritance, means more the dominant character, more the darker color. Okay, so if there is a red color, so it is being taken care by two genes controlled, A and B. So this would be what? Red in color. And the another one, which is all the recessive genes, means would be what? White in color, no color. Always remember the recessive color would be white. Okay, so then we will make what? Gametes of it. This will be this gametes, this will be this gametes. They will do what? Cross, and it will form what? This color. And this color is what? The intermediate. Okay, so this is intermediate. Now if you will make it what? Punnett square, F2 generation. So if you had made the Punnett square, if not, then make it yourself. Here I am just giving a rough idea of it. So AB, capital A small, small capital, small small. Same over here would be there. So capital, capital small, small capital, small small. So over here your phenotypic expression would be what? 4 dominant, 3 dominant, 2 dominant, 1 dominant, and 0 dominant. So 4 dominant, there will be only 1, 0 dominant also 1, 3 dominant, 1 dominant, and this. So your expression would be 1:4:6:4:1. Similarly, you can make what? Its cumulative graph like this. Okay, so this is what one such example which you have to remember. That's all about the polygenic inheritance for now. So the next thing that we're going to be studying, which is super important, is Pleiotropism. Okay, so what is Pleiotropism? Let's just see. Now, Pleiotropism is a phenomena. What phenomena it is? That normally till now what we were saying, that one gene or more than one gene is influencing one character. Opposite of it. What if one gene is affecting multiple characters? This is what Pleiotropism. So what it is in simple words, if we have to write that, when one gene affects many, affects or influence, okay, affects or influence many or more than one characters. This is what Pleiotropism. Okay, in easiest way I have written up. No fancy lining. So how do we do that? To do that, we need to understand an example. Okay, what is the example? For example, a disease called as phenylketonuria. Okay, in disease phenylketonuria, what happened basically in this? Let's just see that and how it's affecting. Now in your body, whenever phenylalanine amino acid is present, okay, so we have what? Phenylalanine amino acid. Now this amino acid, since in our body every amino acid has to be metabolized into certain other form, so normally what happen, under the influence of phenylalanine esterase enzyme, that is what one enzyme I'm writing just now, one enzyme will work and changes it to what? Tyrosine amino acid. This is how their normal timeline is. Okay, this thing has to be changed into this thing in our body, because phenylalanine like that way will do certain problems. Now what happened, this enzyme which is required to convert it, it is defective. Maybe it's not forming or not forming properly because certain gene is defected. Okay, so what happened, the gene required to form this enzyme is defective and unable to produce enzyme, or if producing then it is defective or less common or abnormal enzyme. Hence this conversion is obliterated, means it's not taking place. This is understood now. If the enzyme is not forming, this conversion won't happen. Okay, so this conversion is obliterated. Now what will happen? If the conversion is not taking place, means this phenylalanine which is present, it will stay like this. So if someone will stay like this, then it will do what? It will go and do certain things in the body. So just imagine that if you have a toy and you are not playing with it, you do what? You just throw it in the one corner and it is in that corner only. So means this aniline will also do what? Like, it is in my body. In my body, it is not doing anything. It's not converting into something. Means no one is playing with it and changing it to some other form. So what will happen? It will keep on getting accumulated in my body. Where? In the joints and the cartilage. Because joints and cartilage are the easiest places to get the things done and dump it. Okay, so what will happen? Due to this reason, phenylalanine gets accumulated in the joints or cartilage. And now if they are getting joined over there where they not should be present, they will create what? Problems. Okay, so they causes pain, all those things. Along with that, it affects what? Nervous system, wrinkles of line problems come, facial expression is changed, reproductive system is also exploited. So means what happened? Your brain, facial expression, reproductive system, everything is affected. Means different organs are affected. Means different expressions are coming up. One gene problem which is not making the enzyme is affected brain, facial expression and the reproductive system. Understood this thing beta? So this is what a syndrome kind of condition, which is what? Phenylketonuria. So phenylketonuria is what? A syndrome. Syndrome in group of effects showing, which is far vast off. Understood this thing? So it leads to affects central nervous system, facial expression and reproductive system. Hence, one gene defect affects multiple characters. This is it. This is what? Your phenylketonuria. I hope this phenylketonuria is understood to you. Next we're going to be studying about the lethal genes. What does lethal word means? Lethal word itself means what? Something which is deadly, fatal. So lethal genes are what? Certain genes are there in our, like, because they are normally not present, but with the mutation they came into our population. So what happened? If there are certain genes, they are okay if they are present in their heterozygous conditions. But if they are present in their homozygous conditions, means it will be lethal. I will give you an example. Okay, so what is the lethal genes? Certain genes certain genes, if present in homozygous condition, can or turns out to be lethal or fatal, or it could call as deadly. Right now, what basically happens? So that can even, like, certain genes, if they are not very lethal, like normally what happens, the child dies within the womb itself if they are present. If the child survives and survive for a certain period of an age after the delivery, then we call these conditions as semi-lethal genes. Okay, so means normally such progeny with lethal genes dies within mother womb if it survives for few years, then we call it semi-lethal genes. Semi-lethal genes. Understood this one thing. Now let's see the example of it, which is the most important thing. Example is sickle cell anemia in human. Sickle cell anemia in humans. Basically, what this disease is? This is the disease of RBCs, where the RBCs are sickle shaped, means curved, C-shaped, look like sickle. Sickle is what? A instrument which help in digging the soil. Okay, so it is like a not digging the soil, but to cut the crops. It is like that. Okay, so sickle shapes are there. So the sickle shaped RBCs are there. Now what would be the problems? The RBC surface area will reduced. If the surface area is reduced, means it leads to what? Hypoxia condition. What is hypoxia? Low level of oxygen. So oxygen won't reach to the body organs and causes what? Hypoxia. So let's just come back. That it in the sickle cell anemia. So let's see that. So basically there is a gene called HB, like hemoglobin gene. So basically the chain, normal hemoglobin chain is what? Round in shape, biconcave in shape. But these are what? Sickle shape in the shape, because there is a defect in the hemoglobin chain. What defects, how defects, later we will understand. Okay, when we are doing the diseases, genetic disorder diseases, that will come. Here we are just writing that what is happening. So HBA is normal hemoglobin chain leads to round shape RBC. But if by any mutation, what mutation will occur, we will do later on. But some changes, you can say that this chain forms, that chain change. So what happened? HBS is what? Sickle shaped RBC formed. Okay, now let's just see an example over here that how it can turns out to be lethal. Now imagine that there are what? If the gene are there, two alleles would be there. Imagine that there is a, if I am there and I have what? I am having both the things. If I have both the things, means in my body, both the genes would be present, genes are present, genes allele form is present, means it will make a round RBC also, sickle cell RBC also, and my body is completely fine. No much problems are there. When the problem arises, let's see that. If my wife also has what? The same hemoglobin chain formation. Let's just see that what is happening. So imagine that there is one person with this, means male is this, and female is also having what? This condition. This condition is called as what? Carrier, because we will be what? The carrier of the allele which causes disease. So they are what? Carrier. So this is carrier. Can you see this thing? This thing is carrier. So understood what is carrier. So this is also carrier. So the first condition that will be forming after this gametes formation would be this. So means here both the offspring that would be produced, that would have what? Both the AA, means there will be what? A healthy offspring will be there. So this is what? Healthy progeny. Now other option is what? Other option is we have this, that this can go and join with this one. So here what will form? HBA, HB. So as I said that this would be what? Healthy one, and this is what? Carrier one. Carrier is also healthy, no problem is there. So this is what? Carrier, but totally fine. Okay. And then what happened? This has option to go with this. So that is also what? Same like carrier. So this will also be carrier. These all will what? Survive. Because healthy will definitely will survive. You know now this A means what? Healthy. Normal. Normal thing will surprise. Here the carrier will survive. Okay. What is this? The last thing. That is this with this. Here what happened? HBS, HBS. Means what? Here the disease will occur. Means this is what? Sickle cell. Sickle celled anemia case. And this thing will what? Die. So means one progeny will die. Understood? So this is the case. Here what? If we say that healthy ratio carrier, that is 1:2. Of course 4 offspring come, but one will die now. Okay, so that is what? One will die. So there is 20% chances that if the parents both are carrier, their child can be sickle cell anemia. So we have to find it out whether they are sickle cell anemia or not. And how do we can find it out? By the certain, we have certain ways like amniocentesis and all those things. If you have studied the chapter number 3 of your NCRT, that is reproductive health, you might have understood it. If not, we will do it that at the end of the session. Okay, let's just understood that what is the lethal genes. Lethal genes are those genes which has chances that they will leads to what? Death. Example is sickle cell anemia. In this there are, if the sickle cell alleles are present in the homozygous condition, the child will die in the womb itself. Okay, that is about the lethal genes. After lethal genes, we're gonna be studying about the cytoplasmic inheritance. Cytoplasmic inheritance. Now what happened? In cytoplasmic inheritance, you have to understand that normally your genes are what? Present inside your nucleus. But beyond the nucleus also certain genes are present. Can you name somewhere where it is present? Okay, I tell you. If you studied about the cell chapter, in cell chapter you might have studied two different organelles, that are what? Chloroplast and the mitochondria. In both of them, they have their own genome or the genetic material. If they have their own genetic material, means they will have certain of the characteristics also. Now, these things are present what? Not in the nucleus. These organelles are present in the cytoplasm. So that is what? Cytoplasmic inheritance. Means the things which will be affected by the cytoplasmic genes which are present in mitochondria and the chloroplast, that will be affected over there. So all those genes which are present in the cytoplasm called as plasmon. So first thing we will remember what? Genetic materials present in cytoplasm. Where in cytoplasm? Mitochondria and chloroplast. Is termed as plasmon. That's it. Okay, now let's just see that example of it. This example which I am taking is very simple one. Means now think about it. Who will be the affecting ones? In a sexual reproduction, there is a female or a egg which is the bigger in size and the male is very smaller sperm. Now, if you remember the fact that the cytoplasm is negligible in case of sperm, whereas in the egg, in the female gamete, it is the most abundant things. They contains, if the cytoplasm is there, means the organelles will be present. So means the maternal inheritance concept is this only. Means what happening, beta? That whatever the genes present in the mother, okay, egg, in the cytoplasm, that effects will be shown. Okay, so what it is? Since egg, that is female gamete only contains large portion, okay, only contains cytoplasm, thereby the characters of maternal plasmon is inherited by progeny. Means that in my body, all the mitochondria that is coming up is from my mother side, if we see like that. So this is the feature. I hope this thing is understood to you. Let's give it by an example. So example number 1, there are many examples. One example I'm saying, other we are giving just example, like naming we have to remember. So example is the shell coiling in snail. So beta, you know that snail look like what? Something like this. And it has what? It has a shell like this. Now this shell will be present in the coiled form. So this could be anti-clockwise or the clockwise. If it is a clockwise, we call it dextral coiling is there. If it is anti-clockwise, we call it sinistral coiling is present. Let's see that. We have a mother, we have a father. So the mother is, let's take it, dextral coiling is there. Whereas male has what? Sinistral coiling is there. What it is? Direction. Sinistral means what? Your anti-clockwise. And this is clockwise. Now what will happen? When they will have a baby. Okay. Now, no matter what is there, they will show what? Dextral coiling only. All. Okay, so this will be seen in their cases. They will have 4 baby, we know that, but all of them will have the dextral coiling. Understood this thing? So this is same. If we have to remember the other example, so we have example of stem color of dog plant, or there will be another example that male sterility in maize is also affected by what? The cytoplasmic inheritance. With this, all of the inheritance are over for you. Let's just go towards the next pointer, that is crosses. So certain crosses we have to remember, starting with the easiest one, and then we will go towards the main one which will be there in your board exams as well. So number 1 cross that we're gonna study is the reciprocal cross. Now what is reciprocal cross? Name itself saying that we can change the things will change. That depends upon the cytoplasmic inheritance. So what happened in this cross? If we change the parent, the character will also reciprocally changed. Example, same, this cytoplasmic inheritance. What happened? Shell coiling in snail. Okay, let's take an example, male and female. So if the male, you remember na, we had taken that sinistral coiling was there, and the female has what? Dextral coiling. So all the baby would be coming what? Dextral. But if I took a female, female with the sinistral coiling. I have changed the character. Okay, and the male is with the dextral coiling. Then which character will come? Sinistral. So means here we are not saying that which is dominant, sinistral or dextral. It could be anything which the female resembles. This is the one. Okay, after the reciprocal cord, we have back cross. Back cross means what? That whatever the progeny coming, it goes back and mate, or the cross is made between one of the parents. Okay, the progeny is made to cross with either of the parent. Either of the parent. That is called as what? Back cross. Okay, means for example, just I am writing it over here. So if this is AA and this is what the progeny will form? A. If this progeny I am making it cross with either this or this is known as what? Back cross. Okay, each time whosoever I am doing it, it is what? Back cross. Next come the last topic of today and very important, that is test cross. So in test cross, what will happen? That in this cross, we basically do what? The homozygous recessive is crossed with unknown genotype, unknown dominant genotype. Yeah, dominant means phenotypically it is showing the dominant, but genotypically we don't know, because either it could be homozygous case or the heterozygous case. Let me make you understand. Don't worry about it. So test cross is what? First you have to remember the fact that is, it is a cross between homozygous recessive, means small. Okay, homozygous recessive with unknown dominant genotype. Means what? Heterozygous or homozygous, what it is. Okay, so means we have to find it out what? That whether it is this or this, we don't know. Okay, now in both the crosses, basically what is happening? Basically, it's cross between F1 progeny to the homozygous recessive parent. Homozygous recessive parent. We can say it like that also. Okay, one way. Okay, not the completely correct statement. One way we can say it like that also. So let's just take an example and see that what happens. Okay, so this cross would be what? We are making a cross between a character. Okay, so example, cross between dominant character of erect ears, that is capital E, this both will give the erect E, with homozygous recessive character, that is small e, that is dropping ears in dog. So if you might have seen dog, either they have the erect ears or they have ears like this. So this ears, dropping ear, is a feature of what? It is a feature of a recessiveness. means recessiveness will only be shown if it is both in the small. Erect ear is what? It is a dominant character, means even if single capital E or double capital E, whatever come, it will show. We have to find that which one is this. Whether the, for example, you got a dog like this. Now it is asked that find out whether this dog genotype is this dominant or recessive one. Okay. If we have to do it, what we will do? We will find a partner which is dropping ears. Okay, so what happened? You have a, for example, if, now what will happen? Imagine that you have done this and you found out that all my progeny looks similar. First case. Second case, all will look different. If it is looking similar, if it is looking different, it will tells me whether it is a homozygous or heterozygous. Let's check it. For example, imagine if I have a homozygous condition. Okay, so means my partner would be drooling one, which is a recessive one. Okay, so I have this one, so I am right making it. Okay, let's see that. So I have this one. So case 1, that is dominant. I am the homozygous case. Homozygous. If so, this is the female part. They will make what? They will make these gametes. They will do cross, and you know the when they will make this, let's make the Punnett square. Capital E gametes, small e. So capital E. Can you see that all offsprings in this case are what? Similar looking. All offspring, that is 100%, will be dominant phenotype. So means all my babies, if I would be a homozygous, then all my babies would be what? Same looking, means they will be also having erect ears. But if I would be heterozygous, okay, that is case 2. If I am this, and then there is a cross making. So means options are what? These two gametes. Here the gametes would be this. Okay, let's make a Punnett square in this case. Okay, how it will look? Let's see. So capital E small, capital small. Means here the phenotype is too different. If you see this phenotype, it is what? Recessive. This is what? Dominant. Means 50% of my baby in this case, if I would be heterozygous, okay, it would be what? It would be 50% dominant characters showing, 50% drooling ears showing. So it is what? If heterozygous, then 50% and 50%. So 50 would be what? Dominant character, and 50 would be what? Recessive character. And that's how they will be able to find what? Whether I am what? Heterozygous or homozygous. And this becomes an important thing. I hope this thing is understood to you. Let me just recap you quickly because the understanding is very important. So what happened if I have a erect ears? Now erect ears is what? A dominant character. Even if heterozygous condition would be there, it will be what? This ear, right? Now this ear condition, if it is there, now if someone want to ask me that if this dog genotype with respect to this is what? It is a homozygous or a heterozygous. So if I would be a homozygous, then all my baby, when a recessive dog, drooling dog, female dog would be breed with me, then all the babies would be what? Erect ears. If that condition comes, 100% babies comes this, then what it means? It means that I am a homozygous dog. But if I am this, my spouse is this, and the baby comes both this and this, my babies, means what? I am a heterozygous condition. Means heterozygous conditions, recessive trait will come into equal number. That's the thing. I hope you like this video. If you like this video, give it a like, share it with your classmates, friends, so that they can also be benefited from these topics. You can comment down and let me know that what things you are understanding and what problems you are going through with it. I will try to clarify it. Anything which is not being understood, just comment it down. I will keep on working on these doubt sessions also in between the videos. Well, that's all for today's video. Thank you so much.